Hello everyone, my name is Byeonghwi Lim. I'm very grateful for the opportunity to present my research in NAFRI of Chungang University. I'm currently a postdoctoral researcher in the Department of Animal Science at Iowa State University. Today, I will present our recent work titled Functional Genomics Approaches to Immune Regulation and Adaptation in Livestock. This talk highlights how omics integration can help us understand the immune regulation and adaptation under infections, xenotransplantation, and environmental stress. My presentation will cover five parts: background, case study 1 about infection, case study 2 about xenotransplantation, case study 3 about environmental stress, and finally, summary. So, let me start with the overall background. My research focuses on how livestock animals regulate and adapt their immune systems under different challenges, such as infections, xenotransplantation, and environmental stress. To do that, I use an integrated omics approach, combining multiple data types and time series information to get more complete pictures of what's happening biologically. And ultimately, the goal is to identify molecular signatures that can improve immune resilience and productivity in livestock. Let's start with first case study, which is about Pierce virus infection. The title is Single-cell transcriptomics of bronchoalveolar lavage during PRSV infection with different virulence. Porcine reproductive and respiratory syndrome, which is called PRRS, is one of the most costly diseases in pig industry. It's caused by PRS virus infection, and it mainly infects monocytes and macrophages, but only around 5% of the cells. In those, PRS can lead to reproductive failure like abortion, olivary, stillbirth, mummified and weak piglets. In green pigs, it mainly shows up as respiratory problems, as you can see in the pictures here. However, vaccine efficacy remains limited due to the rapid mutation rates of PRSV strains. Also, host responses and viral pathogenicity exhibit substantial variability across strains. Single-cell research can serve as a highly effective solution for identifying complex immune cell populations. When we look at the tissues, they are made up of many different cell types. For example, in brain, bulk harnessing mixes all those cells together, so we only get an average expression. But with single-cell RNA-seq, we can separate those cells and look at each cell type individually. So this allows us to identify cellular heterogeneity and distinct cell state. In this study, single-cell RNA-seq can be useful to identify pathogenicity-dependent cellular changes in both immune cells from PRSV-infected pigs. We used 36 pigs at 4 weeks of age and assigned them into four groups, including negative, low, intermediate, and high virulence PRSV strains. As you can see in the figure, long samples were collected at 3, 7, 14, and 21 days post-infection, which is called DPI. After isolating BARF from the lung samples, we performed single-cell RNA-seq along with several additional downstream analysis. From the single-cell-seq analysis, we identified over 300,000 cells. These cells were broadly classified into three major categories: myeloid, lymphoid, and epithelial cells. And as you can see in the figure, myeloid category is a majority. Then we analyzed all groups and time points separately to capture distinct cellular responses. As you can see in the middle on the left, the low virulent strain showed almost no changes, but in the intermediate and high virulence infections, myeloid cells dramatically dropped due to the viral infection. This point can be called peak damage point. The key difference between them is timing. The high virulence strain showed this myeloid decline at earlier time point. And in the middle on the right, you can check the viral transcript, which is described like blue color. They showed abundantly before peak damage point. Next, we focused on the macrophage and dendritic cell populations, which account for the majority of the cells, as I mentioned before. The macrophage and DC cell population consisted of 23 subcell types. In the center, you can see two clear patterns, density patterns. The certain subcell types showed shared density pattern across both intermediate and high virulence infections. Red circle. Also, certain cell type displayed clearly separated density pattern between the two groups. Yellow circle. Down here, these plus show 8 major subcell types. Across both virulence levels, we still check a clear drop in macrophages at the peak damage point. But interestingly, SPP1 expressing macrophages increased. You can check the last two macrophage subcell types. And this increase in SPP1 and CXCL14 macrophages was observed only in the intermediate virulence group. Based on their marker gene expression, this population may play a role in supporting recovery as a M2-like macrophages. When you compare the infected macrophages with bystander macrophages, the infected cells showed broad downregulation of metabolic, immune, and cell signaling pathways, including apoptosis. This suppression was even stronger in the high virulence group. As consistent with previous findings, showing that only a small portion of macrophages are infected. The infected macrophages displayed broad functional suppression. These impaired macrophages can be likely, likely to influence bystander macrophages' death indirectly. Based on the comparison between bystander and negative macrophages, bystander macrophages show the regulation of immune signaling at the peak damage point, as well as increased necroptosis and pyroptosis. And these responses were more strongly induced in the high virulence group. As expected, the bystander macrophages showed upregulation of signaling pathways associated with cell death. In conclusions, large-scale comprehensive longitudinal changes were identified in the immune cell composition of BALF with varying virulence levels for PRSP. Especially anti-inflammatory M2-like macrophages can contribute to the lung recovery from intermediate virulence infection. Move on to the second case study, which is about xenotransplantation. The title is Comparative Transcriptome Analysis between Long and Short-Term Survivor after Pig to Monkey Cardiac Xenotransplantation. There's a simple background. Xenotransplantation refers to cross-species organ transplantation, and current research mainly focuses on transplanting genetically engineered pig organs into non-human primates. It's becoming it's becoming increasingly important because xenotransplantation is emerging as the only treatment for patients with untreatable organ failure, especially in heart. RNA sequencing enables comprehensive quantification of transcriptomes by sequencing cDNA fragments and mapping them to the reference genome. So RNA-seq can be useful to identify molecular differences between long and short-term survived monkeys. For the study design, a genetically engineered pig's heart was heterotopically transplanted into a cynomolgus monkey. The monkey survived for 60 days, and we call the POD 60, and the transplanted heart was collected at the endpoint. To match the timing, we also collected hearts from age-matched pigs at the same time of transplantation and at POD 60. Then we performed RNA-seq and differential expression analysis to characterize transcriptomic changes related to survivor. Finally, we compared these data with a short-term case, a monkey that survived only 9 days, to identify the molecular differences between long and short-term survivor. For the In the result, we clearly checked that all groups were well separated. When we compared each xenograft group with the age-matched pig hearts, we identified around 1800 and 1600 differentially expressed genes, which is DEGs, respectively. Look at figure C. Many numbers of overlapping DEGs were identified in Venn diagram, and using only these shared genes for functional enrichment, we found pathways related to metabolism, immune, and cell signaling. In addition, when we separately analyzed the up and down or regulated genes, both sets consistently highlighted immune-related signaling as a central functional category in the D and E. D for D's for geoterms and E's for kegterms. Unfortunately, the E resolution is bad to check. We compare the short-term survived monkey with the long-term survived one. From the Venn diagram, three gene sets were defined. First, short-term survivor specific genes, yellow, long-term survivor specific genes, green, and the overlapping genes is red. Then these gene sets were summarized into CAV pathway terms, which are shown in the left table and right figure. Both groups showed enrichment for heart failure in the red, but the long-term survived monkey displayed unique signatures in immune signaling and fat metabolism. It's green. While the short-term survived monkey showed distinct patterns in cell signaling and neural and hormonal signaling. It's shown in yellow. And when you looked at cytokine signaling, we also confirmed that terms were distributed across the long, show specific and overlapping gene sets, showing diverse patterns. If we take a look at the details, for example, the cell cycle pathway related to cell signaling, we found some important genes involved in heart remodeling and regeneration that may adversely affect the xenotransplanted heart and its survivor period. As shown in the figure, the yellow squares represent short-term specific genes, and each of these genes was clearly upregulated only in the short-term survived monkey, left side of the square gene. We also looked into the cytokine receptor interaction pathway. As you can see in the figure, the yellow squares indicate short-term specific genes, the green squares represent long-term specific genes, and the red squares show the overlapping genes. These gene sets show distinct expression patterns across survivor groups, highlighting that different cytokine-related genes are involved in long-term and short-term outcomes. Functionally, T-cell activation was commonly suppressed in both groups, whereas B cell activation showed a stronger suppression in the long-term survived monkey. Additionally, T cell active addition no. Additionally, we validated several key genes related to cytokine signaling using RT-qPCR, and the results were consistent with the RNA-seq findings, confirming the reliability of our transcriptomic analysis. In conclusions, the heart failure features were shown in common, with remodeling and regeneration specific to short-term survived monkey. Long-term survived monkey exhibited neurohormonal multi-organ failure features and B-cell suppression. Short-term survived monkey uniquely showed chemotaxis and pro-inflammatory cytokine activation. So these findings reveal survivor-related pathways and candidate targets for improving xenotransplantation outcomes. This is last case study, focusing on environmental stress, specifically heat stress in broiler chicken. The title is Integrated Transcriptome Analysis for the Hepatic and Jejunal Mucosa Tissues of Broiler Chickens Raised Under Heat Stress Conditions. Here's a brief background on heat stress in chicken. The poultry is known to be the most sensitive to heat stress among livestock animals because poultry is covered with feathers and has no sweat glands. Like this figure, the chickens showed a range of responses when exposed to heat stress, starting from behavioral changes, then neuroendocrine and physiological disturbances, and eventually a dropping in productivity. As I mentioned in the previous case, RNA-seq also can be useful to identify molecular changes after heat stress in broilers. For the study design, first, 500 one-day-old Ross 308 male broiler chicks were raised for 20 days, and then 200 birds with a narrow body weight range were selected. These selected birds were divided. These selected birds were allocated to 20 battery cages and then divided into thermoneutral, STN, and Hystress HS groups. Raised for 2 additional weeks, and liver and jejunal mucosa tissues were collected to generate RNA-seq data with some phenotypes. The details in the temperature conditions are shown in the below of the slide. The results showed a clear separation between two tissues, brown and green ones. And within each tissue, we also observe some differences between the TN and HS groups. The HS groups are a little bit higher located than TN. In the liver, the 703 around 700 differential express genes, SDGs, were identified, while around 300 DGs were detected in the jejunal mucosa, indicating that his stress had a much stronger impact on the liver than on the intestinal tissues. And we can check only a small number of overlapping genes were found between those two tissues, highlighting their distinct transcriptional responses to his stress. Using DGs from each tissue, functional enrichment showed that his stress, his stress mainly altered metabolic and biosynthetic pathways in the liver. For example, biosynthetic of amino acid, alanine, aspartate, glutamate metabolism, glycine, serine, and threonine metabolism, and carb metabolism. Otherwise, immune and inflammatory pathways, such as cytokine receptor interaction, influenza A, and node-like receptor signaling pathway, were most strongly enriched in the jejunal mucosa sample. For the integrative analysis, DGs from the liver and jejunal mucosa tissues were combined to generate a hierarchical clustering heat map. As you can see here, red square showed groups of genes with strong positive correlations between them. It consisted of around 300 DGs in the liver and around 170 DGs in the jejunal mucosa tissue. Using the highly correlated genes in the red square, we constructed an integrative CAG pathway-based network with the DGs. The strong connectivity, the strong connectivity, the strong connectivity among these genes suggests a tightly coordinated liver jejunum metabolic interaction under heat stress, connecting elevated energy demand in the jejunum with enhanced amino acid metabolism in the liver. In conclusions, for liver, heat stress increases amino acid oxidation and shifts energy metabolism towards fatty acid synthesis, including elevated tryptophan oxidation linked to stress responses. For jejunal mucosa tissue, detoxification and antioxidant pathways were activated. It indicates impaired intestinal integrity and increased oxidative stress under heat stress. For integrated analysis, cross tissue transcriptomics revealed metabolic crosstalk where the liver suffers elevated energy and amino acid demands of the jejunal mucosa. So these findings highlight core metabolic adaptations to heat stress and identify candidate pathways regulating energy homeostasis in broilers. Lastly, the summary part. Simple summary part. First, these works can suggest an integrated omics framework for immune regulation and adaptation. Second, these approaches can be focused on infectious challenge, xenotransplantation, and environmental stress. And third, omics integration can help to reveal molecular signatures that contribute to immune resilience and productivity. Thank you for your attention. If you have any questions, please send me email. Thank you.